The moment you swallow a bite of food, it begins a rapid descent into a dark, fluid-filled environment. Deep within the walls of the gastrointestinal tract lies an embedded network of neurons. This system begins analyzing the physical stretch and chemical makeup of your meal immediately, without waiting for a single instruction from your conscious mind. Biologists refer to this autonomous network as the enteric nervous system, the body's second brain. The sheer density of this system is difficult to overstate. Embedded within the lining of the gut are roughly 400 to 600 million neurons. This visualization shows the gut's total neuron count scaled against the central nervous system. Those hundreds of millions of local cells are roughly equal to the number of neurons found in the entire human spinal cord. Extracting nutrients from food requires precise, real-time management of fluid dynamics, of mechanical pressures, and corrosive acids. If your brain had to consciously regulate these variables, it would be instantly overwhelmed. Of course, the gut does not write poetry, reason through problems, or store childhood memories. It operates entirely outside of conscious thought. Instead, the enteric nervous system acts as a localized supercomputer, dedicated strictly to the physiological processing that keeps you alive. The process begins the second a bolus of food stretches the intestinal wall. That mechanical pressure immediately trips millions of local stretch receptors. Instantly, chemoreceptors also fire. They scan the incoming material, identifying precise molecular signatures like glucose levels, acidic residues, and free fatty acids. These combined mechanical and chemical signals drop down into the first major layer of the enteric network, the submucosal plexus. The submucosal plexus responds by dilating local blood vessels, rushing blood into the area to maximize the absorption of the incoming nutrients. At the same moment, it commands nearby glands to secrete specific enzymes and bicarbonate-rich fluids, tailored to neutralize the meal's acidic signature. The gut operates as an autonomous chemical refinery, adjusting its local environment molecule by molecule to match whatever you just ate. While the submucosal plexus handles the chemistry, the electrical signal penetrates deeper into the muscular layers of the gut, reaching the myenteric plexus. When the myenteric plexus detects the bolus, ascending fibers release acetylcholine, triggering a contraction behind the food. Simultaneously, a descending reflex releases nitric oxide, relaxing the muscle in front. Pinching behind and opening ahead creates a coordinated wave propelling material. These mechanical reflexes run entirely on local hardware. However, the brain in your skull still provides overarching supervision to the entire system. During moments of extreme stress or physical danger, the sympathetic nervous system acts as the brakes. It inhibits gut motility, pausing digestion to redirect energy elsewhere. Conversely, when you are safe, the parasympathetic nervous system presses the gas pedal, encouraging the rhythmic, localized processing of resting and digesting. Digestion operates through an intricate mechanical dance, driven locally by the gut, perfectly harmonized with the body's overall state of alert or rest. The primary cable linking these two nervous systems is the vagus nerve. For decades, it was assumed the brain used this nerve to issue commands downward. In reality, 80 to 90% of the nerve fibers connecting the two systems travel upward, from the gut into the brain. This schematic illustrates that upward flow. The gut uses this highway to continuously report its status, directly influencing the brain's emotional centers and stress responses. A crucial participant is the microbiome. Resident bacteria metabolize food, breaking it into short-chain fatty acids. These metabolites enter the system and interact with enteric nerves, influencing how the body synthesizes neurotransmitters like serotonin and dopamine. Every meal feeds human cells, but it also sustains a microbial ecosystem that actively participates in the neural dialogue shaping human mood. Because of this dense physical and chemical connection, functional gastrointestinal distress is intricately tied to anxiety and psychological stress. This connection is visible at the population level. In incidents like the Walkerton water contamination, entire communities developed long-lasting gut-brain symptoms following mass gastrointestinal infections. Translating these mechanisms into treatments, however, is difficult. While specific microbes can reliably manipulate the behavior of mice in sterile lab environments, human biology is vastly more complex. Despite the rapid growth of microbiome research, there is no simple, off-the-shelf probiotic cure for human mental health conditions. When we pull back from the microscopic receptors and bacterial metabolites, we return to the simple, daily act of finishing a meal. Every bite sets off a silent symphony of millions of neurons, hormones, and microbes. It keeps us alive, it shapes how we feel, and it performs its most complex work entirely in the dark.